Good afternoon. I will present you this study on behalf of our research team, which consists of myself, Dr. Salta and Dr. Koulougliotis. I wish to mention that this work is part of my PhD thesis research project conducted at the Department of Environment of Ionian University, which is located on the island of Zakynthos in Greece. My PhD thesis research is funded by a pre-doctoral fellowship assigned to me by the Hellenic Foundation for Research and Innovation. Let's have an overview of the presentation. After the introduction, I will briefly present the conceptual profile theoretical framework as developed by its pioneers. Then, I will describe the purpose and methodology of our work, followed by the presentation and discussion of the results reached. Chemical analysis is a fundamental concept in chemistry and deals with the determination of the qualitative and quantitative chemical composition of materials and chemical substances. The literature on students' alternative conceptions and thinking about the concept of chemical analysis is rather insufficient. Additionally, different approaches have been needed to assess students' comprehension of qualitative analysis. The conceptual profile framework is a methodological tool that can help teachers become aware of the different ways of thinking in science classrooms and via which students enrich their thinking with more scientific ideas. The conceptual profile framework is based on the assumption that people exhibit various modes of thinking that are used in various contexts. Conceptual profiles are composed of several zones which represent a specific way of thinking about a given concept. For a given concept, heterogeneity in thinking is detected in the population and in an individual. Each zone of a conceptual profile emerges from the study of a particular concept in different genetic domains: sociocultural, ontogenetic, and microgenetic. The sociocultural domain can be explored through historical and epistemological research on the concept. The ontogenetic domain is examined by literature on alternative conceptions, and the microgenetic domain is studied by primary data collection such as questionnaires or interviews. For each specific way of thinking, ontological, epistemological and axiological commitments are identified which stabilize it and distinguish it from other ways of thinking. Constant dialogue between data from each domain is needed in order to avoid bias. Therefore, the purpose of our research is the development of a conceptual profile model of chemical analysis through the identification of the different ways of thinking related to this concept. In this presentation, the construction of the zone related to the way of thinking about chemical analysis as everyday practices is discussed as an illustration of how a conceptual profile model of chemical analysis could be developed. As far as the sociocultural domain is concerned, secondary historical and epistemological literature was gathered using search engines and databases and thoroughly studied. The dataset of the sociocultural domain includes some books about history and epistemology of chemistry, analytical chemistry textbooks, relevant conference proceedings, dictionaries, and research studies on the development of chemical analysis from a historical and epistemological standpoint. We used the same methodology to collect data about the ontogenetic domain. Our literature consists of previous studies on students' alternative conceptions on chemical analysis, on relevant concepts such as chemical identity and substance, and on chemistry in general. In the microgenetic domain, the research study was conducted in December 2022. 44 students were recruited on a voluntary basis from the 11th grade of 2 Greek public high schools. Primary data were collected from students' written responses to open-ended questions regarding the process of distinguishing between simple organic compounds, namely ethanol, ethanoic acid, 1-propanol, and propanoic acid. Students were provided with information about these compounds, including their organoleptic, physical, and chemical properties, as well as their applications, sources, and spectra. An inductive-deductive qualitative approach was adopted to analyze data obtained from different genetic domains that were then combined in a dialogic way. For its different way of thinking, the ontological, epistemological, and axiological commitments were identified using appropriate questions that concern not only the entities that are the endpoints of the analysis, but also the analytical processes that are involved. In particular, data from sociocultural and ontogenetic domains were analyzed and categorized in terms of how someone perceives chemical analysis. These expanded categories served in a dialogic way as a coding framework for the analysis of primary data obtained from the students' questionnaires. As we have already mentioned, our results concern the development of the way of thinking about chemical analysis as everyday practices. We will show the findings by genetic domain. Sociocultural domain findings will be presented in a blue box, those of the ontogenetic domains in a white box with a white-blue border, and microgenetic domain findings in pink boxes. We'll start by determining the ontological commitment. Historical and epistemological literature about the pre-alchemical era suggests that the initial type of analysis is often referred to as art. The main idea behind chemical analysis, influenced by Aristotle's analysis of causation, compares chemical analysis to philosophical analysis in the context of separating what is more basic from what is more complex using any method. Basic analytical procedures, mostly employing human senses, such as isolation and separation, were involved in perceiving chemical analysis as such a straightforward process. In the ontogenetic domain, it is suggested that the complex matter indicated by the sociocultural domain is frequently perceived by young learners as separate classes of stuff which are unique and different from one another. Students' tendency to view materials as objects with characteristics mostly ascribed to objects such as size or shape is another intriguing discovery which influences their thinking about chemical analysis. These claims are supported by microgenetic domain results. The most common students' responses involved basic organoleptic procedures of isolation and separation. Other responses are related to the core idea of analysis mentioned before, which is the separation of what is more basic from what is more complex. Also, some respondents see materials as distinct categories of stuff. However, we did not find any thoughts about objectivization. Therefore, the ontological commitment of the zone of chemical analysis as everyday practices includes views of matter as complex entities or categories of stuff that can be separated into their basic parts with simple processes of isolation and separation using human senses. Moving to the epistemological commitment, in the sociocultural domain, basic organoleptic processes were founded on observation alone, employing the use of human senses. Procedures evolved independently of theoretical concepts, and instinct, skills, practice, and experience were crucial. Analytical procedures were not always carried out in a designated and structured area such as a laboratory or documented. The ontogenetic domain's findings are consistent with those of the sociocultural domain. To be more precise, A significant portion of the data regarding students' alternative use of chemical analysis highlights the importance of using the senses. Chemical identity decisions made by novice learners are impacted by surface similarity and explicit features such as smell, taste, color, weight, volume, texture, shape, hardness, and tangibility. Also, when attempting to separate matter, learners frequently place a lot of attention on attributes that are primarily linked to objects. These assumptions are independent of theoretical ideas and limit students' thinking about chemical analysis to the recognition of these obvious traits. Examining the microgenetic domain, in many responses, the following properties were mentioned: odor, taste, color, appearance, form, and clarity, which students perceive through the processes of smelling, tasting, and seeing respectively. Several students emphasize the use of the senses and direct observation. Some other responses support the socio-cultural finding that chemical analysis is not always carried out through experiments in an organized setting such as a lab, while others accord with the fact that analysis of that kind is carried out regardless of theoretical ideas. It is worth noting that no features linking directly to objects or instinct, skill, talent, and experience were detected. As a result, the epistemological commitment of this zone has been refined in that the basis on which people construct their knowledge about the complex matter and the simple organoleptic procedures is direct observation and the employment of senses, emphasizing the explicit properties of materials independently of theory and not necessarily in a lab. As far as the axiological commitment is concerned, according to the history and epistemology of that period, the application and results of chemical analysis were far more significant than the method itself. The main objectives of analysis were to meet everyday and professional needs related to ceramics and metals such as gold, cosmetics, soaps, leather, fabrics, and natural pigments. Data from the ontogenetic domain shed additional light on the evaluation of the entities under consideration. Learners frequently judge a material by its source or typical habitat. Also, they think that valuable behaviors, purposes, or functions may indicate that an object belongs to a larger category, such as an edible material, a construction material, an art material, a cleaning material, etc. In addition, the similarity with popular exemplars of materials widely utilized in our daily lives, such as clay, plasticine, glue, sugar, iron, or wood, is frequently the only judgment learners make regarding a material's identity. Our findings in the microgenetic domain agree with our evaluations in terms of both the entities and the analytical procedures. Students' responses support our argument about evaluating the utility of the entities analyzed and the procedures employed. Furthermore, we identified some more judgments about the analytical processes. On the one hand, they are simple and easy, but on the other hand, they are not so valid and not so reliable. Therefore, the axiological commitment of this way of thinking is as follows. People analyze only the useful entities for their daily and professional needs. Also, they employ only the useful analytical processes which are evaluated as simple and easy, yet not so valid or reliable. So, our conclusions regard the already presented commitments of the way of thinking about chemical analysis as everyday practices. It is important to clarify that in this presentation, neither details on the development of the questionnaire nor results pertaining to the participants are discussed. In a later stage of the research, the remaining zones will be established and stabilized, and a conceptual profile model for chemical analysis will be proposed. The identified zones may then constitute the basis for probing students' thinking regarding chemical analysis. Here you can see some of the sources used for our study. Thank you for your attention. I remain at your disposal for any further questions.